Hello, everybody, and good afternoon or good morning, whichever time zone you're in. Thank you so much for having me. This is Tom Chi. I'm here in Birmingham at UAB, joining you from Zoom. I wish I could be with you in Wisconsin. I really appreciate the invitation for me to participate and be a part of this community. I want to thank Chris Peniston and the organizers of the meeting for always putting together an amazing meeting, and also to specifically call out Dr. Peniston, the work that you've done to bring an entire community together over now almost a decade to really create something special. I think has been incredibly impactful. And to the NIH folks who have supported this community over the many years to try to drive us to better science, better funding, and in these uncertain times, this community is incredibly critical. So I'm delighted to join you to talk about this topic, accelerating a LUTS research pipeline. As many of you may know, I started out as a K12 scholar many years ago. Before there was CAIRIBU, there was still Chris Peniston, but before there was CAIRIBU, and I myself benefited from many of the interactions at meetings like this one. And so I can well appreciate the really big, powerful, positive impact these types of meetings can have. And so we do have some folks from UAB representing our team there, Dr. Farg and Dr. Taguchi and Dr. Knight and others. So hopefully I'll have a chance to interact with people on the team, if not with me. So I was asked to talk about accelerating a LUTS research pipeline. I got a little tangential for a moment because when I was a K12 scholar, I was a Stone person, and now I've transitioned a lot of my work towards BPH and LUTS. I've moved now from being a single institution researcher to now a chair of a department. And over the time, having been a federally funded surgeon scientist, clinician, and led some administrative things, I've taken some lessons away from how, if I could look back and think about things again and maybe change some of my pathway, what I might have done differently or changed. And so hopefully you'll be able to benefit from some of that. So today's talk will really be about, first, I'll share with you one overarching lesson that I learned that was at the beginning of my journey, and I'll reflect on that now towards the middle part of my journey, and then talk really more specifically about LUTS, and then leave you with some thoughts about community. So the lesson that I learned was really from a LUTS guru, and if folks don't recognize, this is Emil Tanago. So he was the chair of UCSF in San Francisco before it was even a department, and was really the first one that kind of made it into an academic department as opposed to a regional or even community type hospital at the time. It's many years ago. And he managed to recruit an amazing team. And I recall meeting him at a cocktail welcome hour, and I was so excited to meet Dr. Tanago. I'd interviewed with him before I got into the residency, and then when I actually got to interact with him kind of face-to-face in a more casual setting, I ran right up and said, started talking to him and asking about his story. And he said a couple of things, and this one always stuck with me. He said, the most important thing is you have to care about the problem more than the solution. And probably many in this room have heard a version of that advice. And that's really stuck with me over the years. He talked about how when he went to do his fellowship in the UK, coming out of Egypt, they said, go to the library and don't come back until you have a good idea and a good problem. And that's what he did. And he came back with some new ideas. And he ended up becoming a person, one of the people that really kind of created this field of neurourology and understood a lot of the physiology that went behind things that now we know as urodynamic studies and testing and many of the things related to neurogenic bladder. And he emphasized if people think that you're onto something that's not a great idea and they tell you it's a stupid idea, maybe it's a really good idea. You should pay attention to that advice, but perhaps there's something to it because many people are anxious about things that are new or different. So those lessons I think I learned from one of the great gurus of neurourology and LUTS and has really stuck with me over the years. So I'll shift gears and share with you a story of my journey from being a case scholar up through some of the work that we've done over the years. And I promise you, just this one little vignette will be about stones, and then we will talk about LUTS. I do promise that. So this journey started when I was a case scholar as a fellow and a resident. I was working on developing a new model for kidney stone disease. And at the time, we had a patient that came into our clinic and said, I actually think that fruit flies form kidney stones. And we thought they were joking, but turns out they were a fruit fly researcher. And their words we took very seriously. And that took us over time to some mouse work. And eventually, with permission, I'm showing you this, it is a video clip. of a person who is taking their first pill on a clinical trial for a drug that was kind of developed out of our lab. And so that long journey was about 10 years or so, which just finished up with that clinical trial wrapping up about a year ago. Uh, to me, there's some lessons to kind of reflect on to share with you and how we might think about that as accelerating a research pipeline for LUTS. And the thing that I took away from that now when I look back on that journey pathway was that a small idea, or some might have said that's a stupid idea, that turned into a pilot grant, which is what that was meant for. And that led on to a bigger grant, a program grant, got us plugged into the CAIRIBU now community and meeting a lot of the people that are in the room with you today, which led us to different types of work with mice and then a human clinical trial. And so our grant pathway also followed the ideas, but it was always thinking about that problem that grounded us. So when we were always thinking about, okay, what's our problem here? Our problem actually is we don't have a great animal model for stones. We want to solve that problem so that we can translate something to some type of cure or some type of medical intervention that we can use for people with stone disease. So if we just follow that problem and go down as deep as our curiosity can take us, eventually the funding map will kind of parallel. And along the ways, it allowed us to branch off in many different things because we just went deep on different problems. And then rather thinking about how are we going to solve this thing, as we thought about the problem more deeply, it led us in different directions to think about kidney ultrastructure, which led to another R21, ultrasound work, and eventually another P20 grant. So this type of following your problem and caring about that more deeply than the solution helped us to not only just come up with interesting ideas and interesting projects, but also allowed us to define our funding pathway over time. So I'll tell you about that story then and what that pathway was like. One of the things that it led us to was, realizing, well, if we want to do something with fruit flies, and I'm a urologist, and my mentor, Marshall Stoller, that I credit a lot of this work with, is also a urologist, and we don't know anything about fruit flies. Well, probably we should find somebody who knows something about fruit flies. So that idea around multidisciplinary work, when you have a problem and you want to look for a solution, getting expertise outside of your field was a really early lesson that we learned. This is Pankaj or PK Kapahi, who is actually an aging researcher. He wasn't himself aging, I guess we're all aging, but he himself was a researcher in the aging field at a local private institute. But he was a terrific connection that we got to, who was a Drosophila researcher. And as we dug into that space, and that's where I spent my K time, we learned that these little fruit fly stones have been described for many years by fruit fly researchers, but never done anything about them. And when you looked at the homology of the genes making up what's called the Malpighian tubules, that's what these guys are right here. There's a lot of about 80% of the transporters in the genes are similar between fruit flies and humans. So that was the start of thinking, okay, well, maybe we actually have something here. So as we pushed that model, we then try to give it all the things that we said, we don't want to give to stone patients. We dehydrated them, we gave them salt, we gave them high protein. And over time, you can actually make them form these fruit fly stones, what I'm calling out right there. And of course, this is the Malpighian tubule. If you can imagine a fruit fly, they're about 1 millimeter or 2 millimeters in size. So we had to micro dissect these things. But when you did, you could actually feel these little stones rolling around your fingers like actual human stones, but very, very small. And then we did things to try to prove to ourselves that they were, in fact, like human stones. So what I'm showing you here is, again, that Malpighian tubule. So this is the equivalent of the human kidney, and it's dissected out, and here are the stones here. And we bound it to things like alendronate that bind to calcium, and we dyed it to prove to ourselves that they're, in fact, like human stones. And we did all sorts of experiments like that to validate our model. And then we again fed them all sorts of things and knocked down the genes that we know exist in stones. So in stones, cystinuria is a relatively uncommon type of genetically bound stone. And when we did things like high protein diets, they form more stones than the low protein patients, in this case, the fruit flies. And so we were very excited. We said, okay, now we've got a model that works and we can push it to make stones, and fruit flies are amazing and we can do these genetic knockdowns. So then when we went back to the NIH and said, look what we did with your R21 money, the response that we got was, who cares? Now move to something bigger and better. And that's when we thought, okay, well, we should try to get a little bit closer to people, and went to some mouse work. And again, here you are with 2 urologists and now a fruit fly budding expert, and we knew nothing about mice. So then we had to turn to some different expertise. So this is Arnie Khan, who's since passed, but was one of the former chairs of a department over at UCSF. And he really helped us to get in this fly work, or this mouse work. And what we did was essentially took our fly model and then changed that into a mouse model by doing the same gene knockdown. And now this is a cysternuric mouse. And what I'm showing you here is that when you knock down the genes, you can make, in this case, the knockdown cysternuric mouse forms lots of stones. That's what these dark spots are inside the middle of the x-rays of these mice. And we screened many drugs in our fly, and the ones that actually screened up positive to prevent stones, we then put in the mouse. And we happened to get lucky, or maybe got smart and lucky at the same time. And one of our hits was lipoic acid, which seemed to block formation of stones very effectively. So what I'm showing you here is mice treated with lipoic acid did not form stones. And when we quantified that, looking at the amount of stones that were formed, you can see here that the control in blue, as we gave them more days off of drug and then gave them drug, that's in red here, they formed way less stones on drug. And you could turn this on and off by giving them lipoic acid. So if you gave them lipoic acid, they'd stop forming stones, and then you stop the drug and they'd form more stones. So it was almost like an on and off switch. So this is a great hit. And we said back to the NIH, look what we did with your P20 money. This is amazing. And they said, okay, who cares? It's time to go to people. And so that's what brought us to our next funding route, which was we also were thoughtful and strategic about looking at the different funding opportunities that were available, especially in the context of this year. I think this advice to me at the time, I didn't really understand it. Arnie Kahn, the guy I just showed you the picture of, he always said, follow the money, Tom, follow the money, Tom. And as a young investigator, I was always railing against that. I was always thinking, no, I want to follow the most interesting experiment. But Arnie was right. If you are careful and strategic and follow the money, that can help you map out your strategic grant pathway much more effectively. So at the time, the FDA and the NIH had together an RFA out for an R01 for orphan diseases. And the recognition we had there was, gosh, we just studied an orphan disease, cystinuria. We managed to show with a reasonable track record some fly work that led to mouse work. And the mouse work was so effective, actually, in this lipoic acid that we proposed a clinical trial that we give this lipoic acid to patients with cystinuria. And I think that the mouse work was so compelling that we actually got funded right away. So that was our first foray then into clinical trials that were federally funded on lipoic acid. And it was a fairly simple trial. We just had a blinded, placebo-controlled trial, 50 patients, because it's an orphan disease, not a lot of patients with cystinuria in the world. And we randomized them to receive placebo versus alpha lipoic acid, which is available essentially as an over-the-counter nutraceutical. And when we got done with the study, what we demonstrated was that patients actually had less stone growth when they were on lipoic acid and they had cystinuria. I'm showing you a couple of CT scans. Now, these CT scans are people and not mice, and they're standing up here. In the white, I'm showing you stones here, a really big stone here on the left kidney, a smaller size stone on the left kidney, and a medium-sized stone here in the left kidney. And then on the right side, at the end of the trial, so all these patients were on study drug, and then at the end, they all got a CT scan. You can see that the stone shrunk over time. And so we wrapped up that clinical trial over time. The thing that we learned along this way was that when you're facing an issue or a challenge that you want to try to figure out the problem on your own, sometimes you have to reach not only beyond your disciplines, but also beyond your walls of your institution. And so we thought that, we at UCSF at the time took care of about 60 or so cysternuric patients, and we thought they'd be running down the hill to enroll themselves in this clinical trial. And I'll tell you that human beings are way harder to control than flies or mice, because when you tell the fly or the mouse to show up for the experiment, they're there and they're going to be early. And when you tell a person to show up, they're like, well, not today. And so we actually reached out to many friends, many of whom are in the room all around the country, UT Southwestern, University of Washington, so many different places to help to recruit patients who then participate in this clinical trial all across the country. And so that community outreach really made a difference. So that's kind of the story of stones. And as I've said, I promised to only tell you one vignette of stones, and then we'll talk about LUTS. But the things that I think I learned from that journey were, I highlighted a few along the way. Looking across disciplines really can help you to amplify your thinking. So if you're in one silo and you find somebody in another silo, maybe if you put your silos together, you might get to some new and interesting thoughts. And being grounded in this idea that whatever you're trying to work towards, if you can anchor your thinking towards translation, that always kind of keeps you focused. So for us, while we were doing a very heavy foray into flies and into mice, as clinician scientists, we were always centered on, okay, what can we bring back to people? And that always helped us to kind of direct our experiments and to accelerate our ability to get through a story like that. And so in the middle, there are many of the patients who participate in our clinical trial, and we're very grateful for that engagement because it makes a huge difference, but it also allows us to bring our findings back to that bedside. So hopefully you've seen kind of from that journey in a parallel universe, which I know is in one of the sessions, I think later tomorrow on stones. Even in parallel universes like stones, you might learn some lessons that you can garner and then translate across to LUTS. But as I promised, we will move on to LUTS and then talk about that for a bit. As I mentioned, for many years, I was a stone person, a stone researcher, and a stone clinician. I started clinically doing some BPH work. And over time, BPH, if you can't recognize, is everywhere. Enlarged prostates are everywhere. And that is actually one of the great secret powers of lower urinary tract symptoms. It is everywhere. It is ubiquitous, and that is powerful. If you don't know some of the data, so lower urinary tract symptoms covers this big bucket of people who have problems with going to the bathroom, number one, not number two. And it is really not hard to find these patients. So people have studied these demographics for a long time. Depending on which study you look at, this is from a little bit of data from a 2023 paper that was in prostate cancer, prostatic disease. And they said that, 19% of all comers, all patients of all ages probably have LUTS of some sort, and that may be an underreported number. If you look a little bit more narrowly at certain populations, lower urinary tract symptoms seem to increase with age. That makes sense. That's what you see when you observe people around you. So as many as half of patients, men who are the age of 50 might have LUTS, and as many as 3/4 or more, once you get to 70, whether you're a man or a woman, might have LUTS. And the data I'm showing you here on that side is from that paper from 2023, and that's just looking at all genders, all ages, all races, in a pretty diverse population based out of Boston of ages greater than 40. And you can see lots of people have LUTS. And people who have LUTS, many of them will get surgery. So this is maybe from the lens of my bias in that I focus primarily around BPH, enlarged prostates that will oftentimes drive lower urinary tract symptoms. And a lot of those patients end up getting surgery. And what we see over time is that the number of surgeries is going up. And that makes sense too, because the number of aged people is also going up. So probably we're going to be doing more surgery. But the thing I will point out to you is when you think about that concept, that means you've got a lot of patients with a lot of data to be able to do research on who may provide you an opportunity to collect a lot of tissue. And that is very powerful. LUTS by nature is cross-disciplinary. And I'm calling out a couple of people. I think that Scott's here at the meeting too. So Dr. Bauer is an associate professor of medicine, really. He's an internist over at UCSF. So it says associate professor of medicine urology, but he's only by association in urology. He's primarily an internist because internists probably see way more LUTS than any urologist will see in an entire lifetime. And many of them get referred to urology when their LUTS get so bad that they can't be controlled or managed. But just by nature, LUTS is a disease that crosses multiple disciplines. And I'm showing you one of Dr. Bauer's partners in crime, Stacy Kenfield, who is employed in the Department of Urology over at UCSF, but they're in epidemiology and biostats. And so when you think about it from that perspective, here's a disease that actually crosses multiple disciplines: internal medicine, urology. Oftentimes, LUTS can get so bad and affect nephrology. And so just by nature of the disease, you have to talk to people, or maybe I should say you have the opportunity to talk to people outside of your discipline. So, and you can see underneath here, I just took this right off of Dr. Bauer's bio over on the UCSF website. He's been continuously funded for many, many years across many different grant applications and mechanisms. Because when you have the ability to do multidisciplinary work by the nature of disease, there's many different opportunities for you to find funding to support the work that you do. And he's a great researcher. And if you have an opportunity, you should talk to him because he's got terrific ideas and always willing to bounce ideas off of you. In addition, LUTS has a long track record of a federally funded infrastructure. I'm pointing out to you Henry Lai, who's currently a professor over at Washington University in St. Louis, and he just became the AUA Chair of Research for the American Urologic Association as of the middle part of this year, and that's a 4-year term. So he's sitting on a great kind of intersection of research that spans across urology and many different other areas, and he'll be the incoming chair over the University of Iowa Department of Urology. Now, Henry Lai has been a big participant in a lot of these CAIRIBU meetings and outreaches. He's been continuously federally funded for almost 2 decades now and has been the PI of the LEARN network, which is the lower urinary tract symptoms of lower urinary tract dysfunction research network. I had to memorize that several times because I think when they came up with the acronym, it was before there was ChatGPT. And so there's some words missing out of the LEARN network. But there's already a federally funded infrastructure that's gone through many different clinical trials, has collected biospecimens and samples. So LUTS is a disease that already sits on a well-established track record. Now, we invited Dr. Lai to come to UAB to give a talk, which was spectacular. It was inspiring. He managed to talk about the challenges to research in the current climate. And one of the takeaways I had there was that when you are studying a disease and there are many structures already in place, don't reinvent the wheel. So if you've got an idea, reach out to a guy like Henry Lai. Say, I've got this idea. There's oftentimes money there already in pilot projects. There are collaborators kind of ready to go to get you plugged in. And you have access to both data and biospecimens right off the bank. So that's another very powerful way to accelerate the work or the idea that you might have by plugging yourself into structures that are already out there and funded. Now, I mentioned this before, and I'll zoom in on this idea. Ubiquitous patients, when we have patients everywhere, it means we have a lot of data, but we have to approach it in a modernized way. And I like to show this picture. This is back when I was a fellow, when we would do data-based research. Dr. Stoll would be like, okay, all right, I've got this idea about stones. Why don't you go back there and let's look up all the patients. And we'd actually physically go to the logbooks that he'd kept for 20 years and page our way through to try to figure out what patients actually has the stone we're interested in, and then collect the data that way. And that, for many years, is how people have populated registries and done retrospective and even prospective clinical research. Now, in 2025 and beyond, if that's the way you're doing research, and that's where you're thinking about doing LUTS research, we have to move beyond that because the data is everywhere. Annabelle Odisho is one of the prostate cancer researchers and clinicians over at UCSF, who's a wonderful guy, and he particularly sits in the bioinformatics space. And so he shared this slide with me. So this is kind of taken from the concept that if you sit on an EMR or an electronic medical record, like Epic, for example, the data is there. So in the EHR, every time a patient comes in, it generates data. Some of it's structured, which is easy to search, and some of it's unstructured, and you have to make it structured. But there's all this structure that sits underneath the EMR that allows you to extract that data to use it both for clinical purposes and quality improvement, but also for research. So how do you leverage that? Now, this is also a lesson that we took out over time where we developed an automated registry in stone disease. I guess I lied. I was going to talk a little bit more about stones, but we are extending this idea now to LUTS and BPH. An ideal disease registry, unlike a logbook that you physically have to page your way through, captures patient data easily. It's easy to populate. It should be low cost. It should take advantage of the electronic medical record system. And if you realize that patients kind of go through a fairly standardized way of coming through a health system, they get referred in, they have a new patient appointment, they might get surgery, they have a follow-up appointment, they might have a different follow-up appointment. It's kind of that same cycle over and over again. If you can turn that into data at every one of those touches, that can populate a registry, which is very powerful. And because we have so many patients, it's an easy thing to do. And this is what we did both for stones as well as for now BPH, where in our EMR, we actually have click buttons that you do, and that will populate your data registry. And we put it in a REDCap as a HIPAA compliant place to actually store the data. But the important concept here is integrate your touches with your patients into data so you can collect that over time and turn that into a powerhouse for your research. Now, to draft off of that concept, here's a lesson that I learned from, this is Eddie Chang. He's the chair of neurosurgery over at UCSF. Wonderful guy. He was a couple of years above me in medical school, but I feel like he was probably like eons ahead of me in brain power. So if folks don't know, the neurosurgery and neurology department over at UCSF, #1 in the country, #1 NIH funded, #1, US News and World Report, all the #1s. My favorite paper that Eddie wrote a couple of years ago now was he's a language mapper. And so what he wrote in a paper was about if you could put an electrode in this exact right way that he figured out onto a patient's brain who was unable to speak, if they thought a word with 80% accuracy, his electrodes could pick up on that word and form it on a screen. I mean, that's like science fiction stuff. That's like the plane that he lives in. And so many years ago, I had the opportunity to sit down with Eddie, and I said, Eddie, how do you do it? How do you manage to accelerate your research and do all these things? And his comment was, you have to integrate. If you're in a lab and you're a clinician, you're running back and forth between your lab and your clinic and your office, that's not very integrated. So when I do my research, my operating room is my lab. So every patient gets onto an IRB, and then every patient I'm putting electrodes in and I'm mapping them. So I'm not doing mouse work anymore. I'm doing human work now, obviously in very humane ways that are all IRB approved. But integrating my lab is now my operating room. So now when I'm in the operating room, I'm doing research is how he's accelerated his ability to be able to be effective across many different places. And that kind of accelerated my way of thinking about kind of, okay, how can we move our research programs forward? So I think this is probably a typical thing. Most people are consumed by patient care. So there's just patients everywhere. You're just over and over again seeing patients. And so you do a little bit of clinical research because it's easier to do. And you got patients in front of you. Maybe you do some outcome study because it's easy to get some data, and basic research which push way over to the side. Now, if you approach to building a research program with intention, obviously patient care is still important, but think about that as part of your program. And when I think about the research program that we've built and I've drafted off and we're building out a UAB, for example, we think about our research across multiple domains. And within those domains, there are things that we do: biobanking, metabolism, genomics, interventions, all sorts of stuff. And in those different areas, we try to integrate as much as possible. So for example, at an individual level, I've got a bunch of different interests. I like clinical care, I like research, I like education, I like administrative leadership, and we can do clinical trials and basic research and translational stuff across many different diseases. But if I find a couple of themes that I can bring across those different areas, then I can kind of wrap my interests all together. So if an example is ultrasound work, which is a clinical tool, I can use it in clinical applications in stones, in BPH, and we can do research on it and also educate people on it. So I'm taking one interest and then bringing it across the different domains to try to accelerate my way to do research. And you can think about that in LUTS as well. If you've got one theme that you can wrap around many different aspects of the program that you're building or your institution has built, then that might be a way to help you accelerate and integrate. The last couple of minutes, I'm going to zoom in on the idea that the LUTS community is very powerful, and that's a great thing. So I mentioned before this idea of, tying yourself in and taking advantage of the structure that's already built around you. If I widen that lens a little bit, and if you just kind of think broadly, and this is just the start of your meeting at CAIRIBU, as you go through the next couple of days, you're going to see all these different highlights. I mean, CAIRIBU always does such an amazing job of bringing out all the strengths that are already in the community. And that spaghetti word salad of all the grant mechanisms that are over on the left-hand side, that's all out there and represented in this room. People have been very successful grant fundings. They represent many institutions. This is a little snapshot of some of them. And you have the opportunity to find collaborators in many different places that are working on things that might be tangentially related to your interest, whether it's LUTS or upper tract or lower tract disease or anything else. There's always going to be somebody who'd be interested in working with you. And this is the little game I'll put in front of you. And the comment here that senior leaders in the community are happy to help, I think is very powerful. I'm showing you some pictures and I'm showing you some names. And the game that I'm putting out in front of you is that, especially for junior investigators or people who are new to the CAIRIBU community or trainees, go out there this next day and a half and figure out who these people are. And I guarantee you, if you go up to any one of them and said, "Hey, my name is blah, I'd like to learn more about what you do, or let me bounce an idea off of you," I guarantee you they'd be happy to sit down and have a coffee with you, talk about your idea, and share some perspectives as well. There are the names. I guess the only obvious one is Allison Wong. But everybody else, it's a mishmash of names along with faces. So the game here is a scavenger hunt of research leadership. And obviously, this doesn't even represent the amazing brain power that's at the CAIRIBU meeting right now. This is just a subset of them, many of them who are PIs and big programmatic grants. Go out there and find them. That's a scavenger hunt for you, junior investigators and grad students and postdocs who are at the meeting. Have some fun with it and make some new conversations because they're happy to help. I know that because, and I guess I'll give you one of the answers. This is Chuck Scales. He's from Duke. I think he's giving a talk tomorrow. we're trying to elevate our clinical trials over at UAB. And Chuck Scales is a big leader at the Duke Clinical Office of Research, and he's done tons of big multidisciplinary studies across different things, the USDRN, which I think he'll talk a little bit about. And I said to Chuck, I would really appreciate it if you don't mind coming to UAB, bring your smartest brain person and help us think about clinical trials in a different way. And he did. He carved out 2 days out of his schedule. And so I don't mean for everybody to now just go ask Chuck for 2 days out of his schedule because then he won't have enough days in the year. But so kindly, to my point, he came, he brought one of his team members. They had 2 days here at UAB and they talked to us about clinical trials. What are the lessons they learned? What are the things that they observed and what we're doing? And how can we accelerate our clinical trials portfolio? And so the senior leaders that we have in the LUTS community are amazing and they're always willing to help. That's been my experience over now almost 20 years of being a part of this community and being able to interact with many of the people on the slides that I showed you just one slide ago. So take advantage of that over the course of the next day and a half at the CAIRIBU meeting. Just know that people in this community want to help you and want to see you succeed, because the more that we succeed together, the more we succeed overall, the better our science is and the further we go. Now, if you extend that idea around community beyond the walls of academic medicine, beyond the walls of your hospital even, there are a lot of community interests on people who have LUTS issues and want to help you as well. I'm showing you a couple of people. This is Marnix Heersink on the left with his wife, Mary, who gave $100 million gift to UAB. It came into a renaming situation, and they funded a couple of institutes, one in innovation and one in global health, and one to recruit and strengthen the institution. And as you can see, that's a demographic that actually probably has LUTS. So if there's opportunities to connect your interest to people in your communities, to engage new partners, to bring them into your research, into your world, into your work, I bet you're going to find yourself with very engaged community partners, which may open up doors to PCORI work, which may open up your doors to philanthropy and many other things. So think about that from a greater context. And then the last thing I'll say is that from a LUTS standpoint, there are huge opportunities for trainees. There always has been, and this community, this CAIRIBU community, the program grant community, and all the people in this room right now recognize and see that. And our people's success is really the greatest markers. We've been very fortunate. I've been very fortunate to be a part of many people's journeys over the years. Some of our fellows have now gone on to do great things. Ryan C. at the top recently transitioned from Vanderbilt to UC Irvine, and he and Greg Tazian, who I think met at one of these meetings, might not have been the CAIRIBU meeting, but one of these meetings, ended up writing a bunch of grants together and just got funded on a big PCORI grant on stones. So our people's success is our success. So for people who are young in their career, young investigators, trainees, really think about that. Everybody's invested in your success. We want you to succeed. We want to help you to get there. And the better we can do that, the better we all win. With that, I'll leave you with two last slides. The first is, this is actually, I'm in my office right now, and this is the door to my office. When we talked at UAB about what are the things that we want to do to kind of change the world and how do we want to actually think, the mantra we came up with was this idea around dream big. So you might think of an idea yourself that's a little nugget, but if you bring that idea out and share it with others, maybe it'll turn into something like a bigger idea. So if you can dream big, we can probably do a lot in the world of LUTS, in the world of benign urologic disease, in the world of research, and really change patients' lives. So many opportunities exist. I'm excited about the CAIRIBU meeting. I'm sad that I won't be able to be there with you during these next couple of days, but you're going to hear about so many interesting topics. And if it stirs an idea in your head, I bet there's a great conversation to be had in these very rooms. With that, I want to thank you all for your attention. Thank you so much, Chris and the organizer of the meeting, for this invitation. I wish I could be there. We had residency interviews this week, and so I had to stay on campus over in Birmingham. Always happy to answer any questions. Please feel free to WhatsApp me or cell phone text me or email me anytime. Be delighted to connect. Thank you very much and enjoy the meeting.